Hi everyone. Thank you so much for having me here today. I'm Ida. I'm a PhD candidate in Dr. Chicheri's myocardial regeneration lab, and I'll be discussing the project that we've recently started, which is the preliminary study of patient-specific stem cell-derived cardiomyocytes and differences in their responses to hypoxic injury, which is all part of the larger Heart in a Dish study. So I'll first discuss the research context of this work, proceeding to our methodology, some exciting preliminary data that we've gathered, and I'll finish off on some future steps. So starting with some background, we're all, I hope, intimately aware of how heart failure is a leading cardiomyopathy and constitutes a leading cause of mortality and morbidity worldwide. And despite the fact that the standard of care has increased over the past few decades, there's a limited efficacy of drugs for heart failure and other chronic cardiac illnesses, and it's still very difficult to treat these conditions effectively and in the long term. Now, there's a very poor prediction of what drug works for what type of patient. And on top of that, the administered drugs have some undesirable side effects, which in turn lowers patient compliance and reduces the drug's effectiveness in the general population. And this is really in part because during clinical trials, the diversity of the patient population is not really accounted for, and conclusions are drawn from a small homogeneous sample. Now, all of these obstacles in developing proper cardiovascular disease therapies are in part due to the fact that current models for HF and other cardiomyopathies have limited clinical applicability. So the real challenge here is, how do we personalize medical therapies for heart failure patients? Ideally, we would have a sample of patient cardiomyocytes on which to test the responses to various drugs. And here there's mainly two options when it comes to cells. The first are for heart biopsies, which are invasive for the patient and really inefficient because the cardiomyocytes which are collected are terminally differentiated, which essentially means that they do not proliferate in culture. So it would really be impossible to perform a repeatable, streamlined, and high throughput drug test. And the second option is to use immortalized cell lines, which are derived from few patients and really don't represent patient diversity. So once again, these cells might highlight cardiotoxic effects of some drugs, but most of the patient-specific reactions would be completely missed by this cardiac model. And so it really doesn't offer much value as a personalized screening tool for drug testing. Now, in 2006, a lot of the limitations that I just mentioned were resolved with the arrival of iPSCs into the picture. So these cells could be achieved by taking some somatic cells and reprogramming them. And once an iPSC population is achieved, we can modulate different pathways and factors that have already been described in the literature in order to achieve the differentiation of these iPSCs into virtually any cell of our body, including heart cells. So this really offers a wonderful, non-invasive method to obtain cardiac cells from a patient for testing purposes or for disease modeling and so on. Now, before we move on to the preliminary data that we have already collected, I would like to quickly walk you through our process. So we received the patient blood and we proceed to the isolation of PBMCs, which are a subset of blood cells. And then we select further for CD34 cells, which are reprogrammable via electroporation. And we actually use episomal vectors, which consist of a slight variation on the classic Yamanaka factors. And even though the success rate is fairly low, so at 0.07, 0.03%, these cells eventually adhere to the plate. And once that step is done, the cells are officially stem cells. So you can see in the circle, they have a very stem cell-like appearance. And our job is from there on to achieve a pure population. So we kind of cut them out and continue purifying them and morphologically assessing them to create a pure population. And then we're ready to actually proceed to the differentiation protocol for our cardiac cells, which you can see there. And you can see that this is a protocol that takes around 2 months before we're able to achieve a cardiac-like cell at the minimum. So how do we make sure that our cells are of high quality and high purity? Well, the first step towards determining this cell quality is during the stage where we produce the stem cells. So for each sample and cell line that we produce, we actually perform immunofluorescence staining of nuclear pluripotency markers, such as OCT4 and NANOG right here, as well as surface pluripotency markers such as SSEA4 and TRA160. And this is in fact the golden standard for most commercialized iPSC lines. And of course, once we generate our iPSC-CMs, we make sure to stain them both for the cardiac markers to confirm that they're cardiomyocytes, as you can see, right here. And we also stain them with pluripotency markers to confirm that there are no longer any stem cells in culture. And you can see here that there is virtually no staining there, which is a good sign for our cardiac cell population. So far with the Heart in a Dish study, we have established a biorepository bank in our lab that currently holds over 100 blood-derived patient samples. But for today's presentation, I'll really be focusing on 3 cardiac lines that we generated and for which we gathered some data. And the different experiments that we have performed relate to contractility over time, to the metabolic activity in response to hypoxic injury, and protein expression in response to hypoxic injury. So here you go, a video, which is probably the most exciting thing that I have kind of seen in a long time while doing science, of one of our iPSC-CM lines. And I'm happy to say that all of our lines that we've achieved through this process, we're able to start beating, albeit to different degrees. And I'd like to kind of bring your attention to the way the contraction kind of progresses from the bottom upwards, kind of propagating. And it gives a sense that these produced cultures have beating synchrony. Now, our first step was to kind of assess contractility over time in a healthy donor and cardiomyopathy patients. And the idea is that during the differentiation process, iPSC-CMs start beating when they differentiate, and eventually the beating slows down. So here we took a minimum of 5 videos a day for each cell line and counted the beats over the course of 20 days. And we can see here that for the control patient, the plateau kind of happens around day 8, which is consistent with the literature. However, for patient CM1, the plateau actually occurs much later. And then finally, for CM2, the plateau is never achieved, and the beating just seems to be more and more erratic as time goes on. So already here we can see differences even at a baseline without any kinds of injury or treatment. So now that we've done that, we actually subjected the iPSC-CM lines to 24 hours of hypoxia and serum-free media. And here we can see that in the control, surprisingly enough, metabolic activity was increased, which could be a stress response. However, in both the cardiomyopathy patients, the metabolic activity either stayed the same or was reduced when compared to normoxia. And while this is preliminary data, this still kind of indicates or points towards the fact that the cells coming from different patients might respond differently to injury. And it'll be interesting to see if this is mirrored in the patients themselves. And finally, we looked at protein expressions in the iPSC-CMs under normoxic and hypoxic conditions. And on the left, you can see the different cardiac markers, which were all expressed in the iPSC-CM cultures. And you can see that in some, such as sarcomeric alpha-actinin, SERCA2a, GATA4, and connexin 43, they seem to be more strongly expressed in the hypoxic injured cells, which is interesting. And could point towards some things that I can discuss in the question period if you're interested. And we also actually started looking at other markers, specifically some kinases that play essential roles in cardiovascular disease, and specifically the protein kinase C, PKC, which studies have shown is expressed highly in neonatal hearts as well as in heart failure, but have decreased expression in adult and healthy heart cells, which you can see right here. And PKC is also responsible for inducing gene expression or by activating the mitogen activated protein kinases, MAPK right here. And what we see here is very interesting in my opinion. While the control right here has low expressions of PKC regardless of whether it's in normoxic or hypoxic conditions, the injured cardiomyopathy patient lines expressed high levels of PKC in response to the injury. And it's been suggested that inhibition of different PKC isoforms should be a target for drug development to prevent irreversible cardiac injury during ischemic reperfusion in humans. So this is kind of an interesting result that I think we should kind of investigate further. So overall, as a summary, I hope that you saw through our preliminary data that at the baseline, generated IPS-CCMs display different beating patterns, that upon hypoxic injury, IPS-CCMs may display differences in metabolic activity and protein expression, that the study of patient-specific IPS-CCMs provides insights on differences between healthy controls and cardiomyopathic patients. And of course, the future steps, the main future step is that a larger population of IPS-CCMs should be studied in order to draw any kind of significant conclusions, whether it be related to the disease state or perhaps sex differences and other differences that are very interesting to study in the context of personalized medicine. So I would like to thank my supervisor, Dr. Chicheri, who's the principal investigator of our laboratory. And these are all the people in my lab who have helped me tremendously in this work and that will continue working on this project with me. So thank you to everyone involved. And I have no disclosures, and I'd like to state that this research was funded through the Courtois Cardiovascular Signature Program, which is trying to pave the way for truly personalized cardiology at the MUHC. So thank you so much for having me, and I will welcome any questions that you might have.